Thanks everybody for joining us for today's GlycoNet webinar. And we have two very interesting presentations today. The first one is going to be from Professor Robert Ben. And Rob and I have known each other for quite a while as we overlapped at National Research Council. Rob did his PhD at the University of Ottawa, and he then did a postdoc at the University of Toronto before moving on to becoming a research associate at the Stacey Institute of Molecular Sciences at the NRC in Ottawa. He then took an independent research position as an associate professor in the Department of Chemistry south of the border at the State University of New York, Binghamton. And then he was recruited back to the University of Ottawa. And recently, well, not that recently, I guess, but in 2012, he was appointed director of the Biopharmaceutical Sciences and Biomedical Sciences programs at the University of Ottawa. Rob's work centers on rational design of synthesis and synthesis of carbon-linked antifreeze glycoprotein analogs and carbohydrate-based inhibitors of ice recrystallization, which I believe is what he's going to talk about today. So without further ado, Rob, the floor is yours.
It's a pleasure to be part of this series. I think it's a fantastic idea. The timing was perfect, obviously, with everything going on with COVID, since it's difficult for many of us to meet in person. So as Warren was saying and implied, I'm going to be speaking a little bit about some of the ongoing work that we're doing in our laboratory towards the rational design of ice recrystallization inhibitors and more importantly, their uses in the translation of that technology. It's going to be a bit of a kind of a 10,000 foot view talk, just because I want to cover some ground and sort of literally start out and show application all the way through. First and foremost, though, I always feel compelled to disclose this. Of course, my day job is as a professor at the University of Ottawa, but I'm also a co-founder and CEO for a biotech company called PanTHERA CryoSolutions, which is the role of PanTHERA is to translate the technology, the ice crystallization inhibitor technology out of my laboratory into commercial and ultimately clinical spaces. So with that being said, I think all of us can appreciate the importance of cryopreservation and more importantly, we can appreciate the role that it has in terms of delivering functional and effective therapeutics, cellular therapeutics, advanced tissue constructs into a clinical setting. And sadly, despite all the advances we've seen in the last decade or more, we have yet to see a true full market deployment of many of these technologies. And one of the major reasons is, in fact, that a lot of these technologies actually have a shelf life issue, which comes back on cryopreservation, which is still a fundamental problem. And I know that many people aren't aware of just how significant this problem is. I was on a call just a week and a half ago with a large firm in the US in the cell therapy space who were trying to figure out what's happening with the cryopreservation of their products. And so these were experts in the field that are encountering some very basic issues. I think part of this confusion comes about largely because we are kind of conditioned to believe that when we use a cryoprotectant, when we go to freeze something, that we often mitigate or prevent a lot of the cellular damage that's occurring. And that leads to this notion that essentially there is no problem here, and that if one use a conventional cryoprotectant such as dimethyl sulfoxide, and then you get fancy and use a rate-controlled cooling rate, so 1 degree Celsius per minute is pretty typical you see in most protocols, that's sufficient, and it's the end of the story. But in actual fact, nothing could be further from the truth, and we've known this actually for quite some time. Way back, cryobiologists have known this since the late 1960s, early 1970s, when people like Peter Mazer and others have extensively studied the freezing effect of cells. And so this is some data from Peter's earlier work, and basically it's showing that every cell type has a unique cooling rate. So one particular cooling rate is not going to work. It's also interesting because even when one uses a cryoprotectant, it shows that recovery of the cells is never 100%. So we always take a hit, and the hit we take in terms of cell loss or cell death is dependent upon the cell types we're looking at or we're trying to cryopreserve. It's also interesting to point out, and I'll say a little bit about this in the next slide, that if we're off of the optimal cooling rate, either too slow or too fast, we experience significant cellular injury and damage. And this is the basis of something called Mazur's two-factor hypothesis. And in essence, what it tells us is that when we're cooling cells slowly, which is typically what we like to do, we get the best response then, we're allowing the cells to dehydrate. And while they're dehydrating, that's preventing intracellular ice formation, which usually is extremely detrimental to cells. After thawing, we do incur osmotic effects and toxicities associated with that result in mechanical stresses. So as a result, we experience cell loss or cell death. Of course, if we go too fast, as I've already implied or mentioned, we do not dehydrate the cells and we get intracellular ice formation, which has largely been regarded as being lethal to the cells. And so we try to avoid that and use cryoprotectants. Now, if you're paying careful attention, you're maybe sitting back and saying, well, the data that I showed you earlier was early 1970s, and certainly we've done better than that, and given that it's now 2020. Because we have a lot of these highly engineered cryopreservation solutions, things like CryoStor, et cetera, as well as some of these sort of more complex high molecular weight polymers. But yet, when you look at data still, what you see is that the post-thaw recovery is highly variable across various cell types. And even using these engineered solutions, we still fail to recover 100% of the cells that we're cryopreserving. And that's really sort of the crux, if you will, for it's an important issue to deal with in terms of cell therapies and regenerative medicine applications, obviously, that's going to allow for translation of these technologies into the clinical settings. But unfortunately, that's not all the bad news, because just after 2010 or so, we started to see a lot of papers like this appearing, where groups have been showing that while we cryopreserve various cell types, whether those are MSCs, HSCs, T cells, whatever it is we're looking at, we may thaw them and take a hit in terms of post-thaw viability, but the cells that we're looking at initially, immediately after thawing, look happy, they look fine. And that's largely because we're using probably the wrong indicator of post-thaw viability, which is usually membrane integrity. And studies have been, increasing number of studies have been showing that, in fact, when we cryopreserve these cells, we follow those cells after freezing and find out, in fact, that they have compromised functional capacity, which again becomes an issue in terms of cell therapies and regenerative medicine when these cells can't differentiate or can't renew in the case of a progenitor cell. So all that being said, I don't want to oversimplify the issue of cryoinjury, because there are many mechanisms which can and do occur during a cryopreservation process or protocol. But it's important to take home or realize that the majority of cellular injury that occurs when we freeze a cell or a tissue, for example, comes about as a direct result of the uncontrolled growth of ice. And that's really the fundamental problem. And in my laboratory, when we talk about uncontrolled growth of ice, we're really referring to this process of ice recrystallization. And simply put, if we look at the images on the top row, top left, you see that we have a frozen sample here. There's no cells present in this sample. It's just straight ice. But you see there's a lot of small individual ice crystals under magnification. And the catch with this is that when that sample is stored, whether that's -50, -80, -200, whatever temperature you're storing at, the ice crystals undergo a dynamic reorganization. So we get this recrystallization process occurring, whereby there's this drive to form larger crystals. And it's these larger crystals that are largely responsible for a lot of the cellular damage that we see when we thaw those cells. So the ideal solution to this actually is to control that process and actually shut it down such that when we freeze, we start with small crystals and we keep crystal size small. And we do this using what we refer to as our ice recrystallization inhibitor technology. And it's important to point out that conventional cryoprotectants, things like dimethyl sulfoxide, glycerol, PVA, et cetera, these actually do very little, or in some cases, nothing to control ice crystal growth and size. They're functioning via different mechanisms and exerting sometimes a small cryoprotective effect. So this area, this isocrystallization inhibitor idea is what we've been working on for probably the better part of the last decade or so. Our lab's been quite successful in developing this platform technology, which can be applied to a number of different systems. And we've looked at many, many different model systems, some with clinical relevance, initially starting with human red blood cells. We've moved to cardiomyocytes, endothelial cells, iPS. I'll talk about those a bit today, various immune cells and progenitors. And even more recently, we're doing some tissue work, actually using this technology to cryopreserve tissue grafts for burn patients. The technology offers a number of advantages. Of course, reduced toxicity. It protects against transient warming injuries, which is something that many people that cryopreserve samples or bank samples don't fully appreciate. We can use slower warming rates instead of the traditional fast warming rates because as a result, we're controlling isocrystallization, so we can move slowly through those temperature zones or zones of transition. Of course, we control extracellular ice growth, but also intracellular ice growth. And I'd like to just kind of pause for a minute and talk about that because, as I mentioned in the slide with Mazur's two-factor hypothesis, we're seeing largely that intracellular ice formation is usually almost always lethal or fatal to cells. And we published some work recently that has shown that our ice recrystallization inhibitor technology is very effective at controlling intracellular ice formation. So what you're looking at here are actually human umbilical vein endothelial cells, or HUVEC. And the image on the top left, we have a number of different nuclei which have been stained with cyto13. And then we've frozen those cells to about -40 using a very high fast cooling rate, which normally you wouldn't do, and in order to trap water inside those cells. And so what you're seeing is this kind of cobblestone effect, and these dark areas are actually intracellular ice. And what's interesting is as we then warm that sample under the cryo microscope and control, take video and take images, what we see is that from -40 to -20, we start to undergo a transition where we see recrystallization occurring where there's larger ice crystals in a fewer number. And as we warm through to -10, we see that process continuing. And then eventually, after thawing, we stain those cells using something like PI for membrane exclusion. And we see that those membranes are compromised there by their dead cells. So this is not surprising. I mean, we're just using a saline solution, no cryoprotectant to do this work or to show this. In contrast, though, when we use one of our really sort of second or third generation ice crystallization inhibitors, you immediately notice significant difference when you look at those that cobblestone effect. So here we have a much larger number of ice crystals, very, very small crystals. And more importantly, during that transition from -40 to -20, we see very little change. The sample is stable under these conditions. And then ironically, when we warm this and thaw this completely and then image those membranes, we see that those are actually intact. So this is pretty significant because it's changing how we view the two-factor hypothesis and challenging us to think differently about whether intracellular isocrystallization is in fact always fatal. In fact, it seems like we can actually control that, and if we control where it's happening and when it's happening, we can achieve a situation where it's not fatal to cells. So as I mentioned in one of the earlier slides, we've done a lot of work in terms of looking at different model systems, demonstrating sort of the efficacy of this technology, the isocrystallization inhibitor technology. And one of those areas has been in actually a recently funded GlycoNet grant in collaboration with my my collaborators at National Research Council of Canada, using induced pluripotent stem cells, or iPSCs. So NRC has large banks of iPS cells, which have been derived from human amniotic fluid. And these cells are pretty significant and featuring prominently in the media and the scientific literature over the last decade or so, because from those cells, we can push them into various cell types that are useful for drug screenings, 3D bioprinting, cell-based therapies, et cetera. But one of the biggest issues is that you get low post-thaw recovery with this cell type. And there's a lot of different procedures and protocols, if you will, homebrews out there for the cryopreservation, which makes it really difficult to adopt something that's going to be effective across the board. So our objectives in this initial grant were quite simple. We were going to basically take the commercially available protocols, which NRC was using, and supplement those looking at the ice crystallization inhibitors. In the early part of that work, we started to look at essentially 4 different ice crystallization inhibitors against a control, which in this case was mFreSR, which is a commercially available solution from stem cell technologies, which is used to preserve various pluripotent cells. And I want to draw your attention actually to this particular compound. This is an aryl aldonamide, which we use in our lab, which has been extremely effective in cryopreserving many different cell types and has a number of different applications. It has what we call an IC50 value, which is really a measure of how effective it is in terms of cryopreservation. So it keeps crystals really small. So the lower the IC50 value, the smaller the crystals are in the sample. And what you can see when you compare, so post-thaw viability normally is about 40% using, give or take, using mFreSR as the control. But when we supplement with the ice crystallization inhibitor, in particular the 2FA, we're getting almost a 2-fold increase in post-thaw viability, which was pretty exciting actually in the early stages. But again, that's just the beginning, right? Because we're really interested in looking at how these cells function, not just being able to increase post-thaw recovery. But nevertheless, very encouraging results, which were initially and still are unoptimized right now. One of the first things we did, though, sort of looking towards functional capacity and assessing how well these cells could actually behave, was to look at toxic effects, right? And so the first 24 to 48 hours after cryopreservation are crucial. The cells are coming out of cryopreservation. They're extremely stressed. Often we see lots of toxic effects, some of which are actually due to the cryoprotectants that we're using. So here I'm showing you a bunch of images, basically, of iPSCs 24 hours after thawing, and the real purpose of this is to compare the control mFreSR to the 2FA. So some of these other IRIs, we saw some other deleterious effects, and so we didn't continue with those, but we did continue with a 10 millimolar 2FA, and looked at various markers of pluripotency, such as TRA181, for example. One of the functional measures that we really focused on, though, was really the proliferation capacity of these cells after they came out of cryopreservation. So there's a few things going on this graph. I'm not going to dwell on it for too long, but first and foremost, what you're looking at here, so these data are obtained using IncuSite, which allows us, essentially, it's a camera inside the incubator and with our cell plates, which allows us to look at confluency as a function of time. Real-time confluency. So what you're looking at here is the blue line, which I'm tracing now, which is really the control mFreSR after thawing. And you see that the cells come out of cryopreservation, they start to do well, and then they start to proliferate. If we look at, oftentimes when people do cryopreservation of pluripotent stem cells, they're using ROCK inhibitors, which is added in during cryopreservation. The role of this is largely to stabilize the cytoskeleton. So when we add the ROCK inhibitor, of course, we see that we do better, as one would expect. But it's interesting, if we just add the ice recrystallization inhibitor in the absence of the ROCK inhibitor, we see that the results are virtually the same, which was really encouraging, because this is a proof that we can actually remove the ROCK inhibitor from the cryopreservation process. And as you would expect, and as we hoped actually, when we use the IRI with the ROCK inhibitor, we have superior proliferation capacity. So this is really encouraging and exciting. But again, it was just the beginning, because we had to start to study sort of the longer term effects of this and look at other measures of functional capacity for these iPS cells. One of those would actually was to look at various transcriptional activators such as OCT4, SOX2, do the appropriate stainings after successive culturing and freezing and thawing cycles to show that the cultures would remain stable and there were no surprises. And indeed, preliminary data indicate that. So we see virtually no difference from the control treatment to when we add the ice crystallization inhibitor in. But the gold standard for this type of work is really the teratoma xenograft model, where we take the iPS and those are transplanted into immunocompromised mice. We grow the tumors, the teratomas, which by definition need to show all three germ layers in terms of development. And as you would expect, when we look at the control iPS cells frozen without the ice crystallization inhibitor, we expect to see, of course, ectoderm, endoderm, and mesoderm, which we see. When we compare that to the ice crystallization inhibitors that have been frozen, the iPS cells which have been frozen with the ice crystallization inhibitors, we see a similar result. Again, attesting to the fact that these are in fact genuine iPS cells. We haven't incurred any sort of unusual or unexpected changes or deleterious effects. So they're actually functional. But as always, we want to kind of continue on and push this and look for other ways to demonstrate the efficacy of this technology. And one of those, which we've done recently, was to look at using deriving human neurons from iPS. So this has been around for quite some time. Obviously, it has this is important in terms of disease modeling, drug discoveries, various cell therapies. Indeed, we're seeing more and more papers appearing almost on a daily basis where people are looking at being able to repair neurological deficits and brain damage, et cetera, as well as I mentioned earlier, modeling for various brain diseases. So these cells are really important. The catch, though, is that the human neurons tend to grow extremely slowly and take a long time to become functional relative to many of the rat models or the rat neurons. So what we decided to do was take the iPS cells, which were previously frozen, take them, actually differentiate them, induce them to become neurons. And as you can see, I mean, this is pretty straightforward to do. We used a commercially available differentiation system to do this. You need to be extremely patient though, because as you can see, based on this timeline, we're talking typically 2 to 2.5 months actually to culture these and get neuroprogenitor cells and neurons. After that time, what we typically do is then we froze those cells down. And we decided in this study to use essentially a control. In this instance, we used CryoStor 10 commercially available cryopreservation solution available from BioLife Solutions as well as Stem Cell Technologies, and compare that to the effects when we have the ice crystallization inhibitor present. So initially we did this, we used conventional freezing protocols. I can talk about that afterwards. It's pretty straightforward stuff using a rate controlled freeze, et cetera, in cryovials. And when we assessed post-thaw viability, looking largely at membrane integrity, which again, we need to be cautious of, right? Because just because a cell maintains its membrane during freezing doesn't necessarily mean it's functional. We really see no difference. This has been done 2 to 3 times now, so we're pending on the stats analysis for this. But nevertheless, I think we can conclude from here that at best we're seeing similar post-thaw viabilities in terms of these neurons, anywhere between using 10 to 5 millimolar of the ice crystallization inhibitor. But as mentioned a number of times already, we're really interested in the functional capacity of these cells. And the way to actually assess functional ability of neurons is to use multi-electrode arrays. And so in this technique, what we do is we culture neurons in about an 8 by 8 millimeter grid, on average between 56 to 60 electrodes actually in each grid. So here's an image of what that would look like. And we culture our cells on these grids, and these electrodes allow us to determine electrical activity. So what we do is we culture those neurons, and we essentially follow their maturation over time. And the measure we're using for maturation is electrical activity and synchronicity and firing of the neurons. And this is typically what you see when we look at the CryoStor control. So what you're looking at here in these rasters is really on the y-axis is the number of electrodes or channels we have. And each of these is about a 20-minute measurement on day 20, day 27, and day 42 in vitro. And so each point represents a firing of neurons. And so as you can see, these neurons are extremely slow, even out to 42 days to mature. They're not really functioning as a network. And that's pretty typical again, right? Because these are derived from human cells. And so as a result, human neurons are very, very slow to develop compared to rat ones. But when we look at adding the ice crystallization inhibitor, we see a dramatic difference in maturation time as a function of synchronicity and firing. So at 27 days already, we're starting to see a lot of synchronous firing of the neurons, which is continuing into 42 days. Just to compare now, I'm going to put these both on the same slide. So on the top, we've got day 27 and day 42 with CryoStor 10, neurons cryopreserved with CryoStor. And in the bottom, we have neurons that have been cryopreserved with the ice crystallization inhibitor. And you can see just how significant that difference is. We've gone out to 132 days using MEA measurements, and you can see that the CryoStor control, actually they do start to catch up, they do start to do better, and we see synchronous firing, but still well behind the 2FA, the ice crystallization inhibitor. So again, this is another example in a clinically relevant model where inhibiting the growth of ice when we freeze a cell sample or a tissue sample is going to be significant and yield not just, in many cases, increase in post-thaw recovery, but yet also increase functional capacity or protect that functional capacity. So we're in the process now of continuing this work, even amidst some of the slowdowns with COVID. We've been doing a lot of the drug testing where we treat these neurons in extensive cultures with various agonists, antagonists, look at firing and opening and closing of various channels. That's part and parcel to this type of work. We've also done some basic staining. In this case, we're looking at beta tubulin 3 and GFAP, which are both indicative of what you expect to see when you look at actual real neurons, staining in both red and green. And we've also looked at MAP2 and NIR-N, which are also, again, indicators of mature functional neurons. And we compare these images for the CryoStor treated neurons that were frozen and then the ice crystallization inhibitor neurons. They're virtually identical, as we would expect, except that neurons been cryopreserved with the ice crystallization inhibitor mature at a much faster rate and would be ready for various applications or for research applications, drug screenings, et cetera, at a much earlier stage. So I'm coming up on my time. I want to sort of just recap, and I hope I can leave you with the impression that this is a really exciting technology. It's an emerging technology that can be applied across many different cell types and tissue types. There's still work to be done. We've been successful actually at pushing some of this into a commercial market. That's the role of PanTHERA. But all of this work actually is really attributed to a very talented team, my research group. And the IPS work that I was mentioning earlier was really done by two graduate students, Karishma and Salma. And all of the neuron work that I was describing was really done by Salma, even amidst all of the slowdowns with COVID. So I think at that stage, I think I'll stop there and answer any questions if anyone has.